Hey everyone, today we're gonna think about gene isolation and manipulation technology. So before we go into more of the details of today's class, we're gonna first think about the word recombinant and in the use of recombinant DNA versus recombinant frequency. So when we talk recombinant DNA, we're talking about the technique that is used to generate different hybrids of the molecules and consist of DNA from two different sources. So recombinant DNA is essentially how techniques that you generate these different hybrid molecules from different sources. And in this case, recombinant typically refers to the artificial joining of DNA from different sources. Okay, so how is this different from recombinant frequency, what we've been talking about half of the semester? So recombinant frequency is used to describe statistical likelihood that two loci in the same organism will assort together or separate during meiosis. The recombinant word here refers to the combinations of loci that are different from the parent itself. So recombinant frequency is the word that we've been using to see what is the likelihood of two loci assorting together or separately depending on what's happening. Right? Like linkage and all that good stuff. So that's recombinant frequency. Recombinant DNA is the techniques that we're going to talk about. So what is recombinant DNA technology and how is it used? So essentially recombinant DNA, as we said, it's joining a variety of DNA molecules, and it is produced by artificially joining DNA, and these are always from different sources and typically not found together in nature. So this is just an example. We have a plasmid from a bacteria. We have that. So we'll talk more about restriction enzymes a little bit later. But these restriction enzymes actually cut open at specific sequences. And then these guys, so another target gene or something else comes in, we get that in. So we have the target gene coming in, we have it be like, okay, now we have our plasmid opened up and our target gene coming in. What do we need to do? We need ligase, right? What did ligase do? Ligase joined nick DNA, right? So if there's any breaks or any gaps between the DNA, they kind of put it together and they seal the openings between each other, right? So that's essentially how recombinant DNA ends up forming. So essentially, the recovered copies of the DNA molecule itself is called clones, right? These clones are typically used to study structure and orientation of DNA. And recombinant DNA technology is essentially used to isolate, replicate, and analyze genes. So whatever happens, you kind of just get one gene. We see how that works and goes like, ooh, yay, we have this one gene, like example, the insulin gene. We're like, okay, what does this guy do? We're trying to figure it out. So we take a plasmid that is compatible with this, and then we end up using other techniques to see how this expresses itself and so on and so forth. This is just a way to allow you to kind of focus on the gene itself, right? Why is cDNA or complementary DNA made only from mRNA and not also from tRNAs and ribosomal RNAs? Why is that? So complementary DNA is made from mRNA because it ends up with so it uses poly T primers. So it's essentially a chain of T's, right? That is the primer that they use to start off reverse transcription, which is essentially the process that we're going to be talking about here. So to transcribe the reverse transcribe this, so it always starts off with a poly T primer to start off the DNA strand synthesis to begin it off. So to bind, so for the poly T primers to actually bind, you need a polyadenylated strand, which is essentially a poly A tail, which is essentially a chain of A's, okay, adenines. So in this case, is you would end up, so typically the only ones that have a poly A tail are mRNA strands, okay? So that is why a cDNA is made from mRNA, because the polyadenylated mRNAs will be the only ones that actually bind to the poly T primers that are used in this process. Okay, so next, so again, why is ligase needed to make recombinant DNA? What would be the immediate consequence in the cloning process if someone forgot to add it? So ligase, as we know, we need it. It's an essential enzyme within all cells that actually seals any breaks in the phosphate sugar backbone of DNA. So in this case, with replication, you know that it joins the Okazaki fragments to make sure that the strand is continuous. So it joins those fragments, right? In cloning, it's essentially to bring in, so it's essentially bringing in the vector and the plasmid. So it's trying to seal those guys together, right? So if we did not have ligase, we would just have the clone DNA and the vector itself just not sealed together. We would just have fragments flying around here, right? I hope that makes sense. So next we're going to talk about polymerase chain reaction, also known as PCR. PCR involves 3 steps. The 3 steps include denaturation, primer annealing, and extension. So these steps are pretty much repeated over and over using a thermocycler, and it allows you to amplify DNA exponentially. And the DNA strands are doubled in each cycle. New strands along with the old strands serve as templates in the next cycle. So this is essentially how it was. So you have a, so to clarify one thing about PCR is that it is a way to, it's an enzymatic process that actually focuses on a certain region, your desired region of the DNA, to amplify those guys out. So you're making copies of a specific region of your DNA, of the DNA itself. So we have a template DNA, we have our set that we want. We're like, okay, let's do this. It goes like a, so it doubled, right? 1, 2, then it doubled again. So essentially it's like a 2 to the n, where n is essentially the, so the number of copies would equal to 2 to the n, where you would just go with the number of cycles as to how many copies you would end up with. Okay, so before we get to a problem that involves talking about like the math of that again, let's talk about what's actually occurring in these stages. So explain what is happening in the stages occurring in a typical PCR reaction at these temperature ranges. So the first one we have is 90 to 95 degrees Celsius. When you have that, that is the temperature where our DNA is denatured. It's like it does it, all the hydrogen bonds between them break and it just pulls apart and it stays apart to turn it into single strands, because you want them single stranded to actually start the process of PCR. Then you at 50 to 70 degrees Celsius, you have the stage of annealing. Annealing is lowering the temperature. So this temperature range allows primers to actually bind to that denatured DNA, and while still keeping the distance between the 2 DNA strands. Next, so we have 70 to 75 degrees Celsius. What's happening there? That's when the DNA polymerase is allowed to come in and be like, oh, yay, we're extending stuff. We're going to add all the nucleotides, woohoo, to the 3' end of the primer itself. So denaturation is at 95 degrees. It's to make single-stranded DNA. Annealing is when the primers come in. And then extension, as the word itself means, it is extending that DNA molecule itself. It is allowing replication to occur. Okay? So this is a question about how we would do the math itself. It is one to think about. In the PCR process, if we assume that each cycle takes 5 minutes, how many folds amplification would be accomplished in 1 hour? So what do we know? We know that DNA strands are doubled in each cycle. and 2 to the n. So essentially the amount that comes in is the number of cycles, right? So the amount of, sorry, let me reword that. The number of cycles is, my apologies. The amplification amount is equivalent to 2 to the n, where n is the number of cycles, right? So, and in this case, we also know that each cycle takes 5 minutes, and each cycle doubles the DNA. So in 1 hour, so we have an hour. In 1 hour, there would be 12 cycles, right? Because it's 60 minutes divided by 5 minutes, because the 5 minutes is what takes for one cycle. So that's how you get 12 cycles in 1 hour. And then you would put it into the 2 to the n, which gives you 2 to the 12th. And that's how you get 4096 fold amplification. I guess I can't, okay, amplification has just been not my word today. Anyways, that is how you get at 4096 fold amplification. I hope that makes sense. If you guys have more questions on this, please let me know. Okay, so let's continue. What are 2 ways to synthesize DNA inserts for recombinant DNA in vitro? So to do this, we have 2 different ways. The first way would have been using PCR to amplify a certain genomic sequences in vitro. So that's one way you can do, you can synthesize this situation. And then another way is to copy the mRNA sequences into complementary DNA using reverse transcriptase. So this guy going backwards from mRNA back to the DNA part, right? Okay. So restriction enzymes. We talked a little bit about it earlier, but essentially it is produced by bacteria as a defense mechanism. So it kind of cleaves at certain spots when a foreign DNA comes into a bacteria or something else, and it is essentially enzymes that cut DNA. Since like the name restriction enzymes, it specifically binds to a recognition sequence, or called the restriction site, and it cleaves it to produce restriction fragments. And this enzyme can cleave at both strands of DNA. Okay, so when it's digesting it. A palindrome, on the other hand, is essentially with symmetry. It there is symmetry between these guys. Oh, yep. So restriction enzymes end up cutting DNA in different patterns, depending on what type of restriction enzyme it is. We have sticky ends, blunt ends. Oh, sorry. So these are like sticky ends where it's like Oh, no, sorry. So these are blunt ends. It ends up with overhangs and, oh, wait, no. This ends up with a double, okay. So blunt ends end up with double-stranded ends. So it kind of cuts right in between. Whereas blunt ends, it kind of goes like one section here, one section there, and it just goes that way with sticky ends. Okay. Why does DNA migrate to the anode or the positive pole? DNA moves towards the positive pole in agarose gel electrophoresis because it is negatively charged. So when we look at gel electrophoresis or any other methods that involve moving molecules by their charge, in this case, you will have DNA go from negative to positive because it moves that way because the DNA itself is negative, right? Negative attracts to positive, right? Like if you put it backwards, it will just go straight out of the gel. So don't forget, always runs to positive, okay? Because labs, many times we've had issues. We don't need our DNA running away outside of the gel. We want it pretty like this, right? So always remember, gel electrophoresis, any kind of methods you use to look at DNA, make sure it is the current is running from negative to positive, right? Okay, good. Remember that. So before we get into the next part, the question would be based on how are dideoxynucleotides or DDNTPs structurally different from deoxynucleotides, right? So essentially dideoxynucleotides actually lack this. So remember how in our sugars we have this hydroxyl group, right? This is the main one for deoxyribose. So in this case, in DDNTP does not have the hydroxyl group here on the 3' carbon. And how does this structural difference end up terminating DNA synthesis, right? So essentially, without that hydroxyl group, you cannot continue synthesis because it cannot form a phosphodiester bond without that 3' hydroxyl group. So that is kind of how Sanger sequencing is occurring. So as always, DNA, it's like a DNA replication process, right? It's sequencing from 5' to 3'. So 5' to 3' going this way because we have our 3' end, we're sequencing that way. So our sequence is going from 5' to 3', and each time, so these different colors represent the different dideoxynucleotides, so DDNTPs. So those guys, whenever they are incorporated, they end up getting stopped, right? Because they cannot synthesize any further. So this guy comes in, oh, we have this entire sequence where it's like G. It comes in whenever one is incorporated, you have that chain end at where it is. It doesn't necessarily follow this order in when they kind of end up stopping or getting incorporated. It can be very random. However, when we do like the sequencing part itself, it's actually separated by size. So that's how we know that this is the order of our sequence. So why are DNA fragments of different length and all ending in a residue that is synthesized? So as DNA is synthesized in the sequencing reaction, the enzyme will randomly insert either a DATP, which has the hydroxyl group, or the dideoxy ATP across from the T residues. So if dideoxy ATP is selected, that chain will terminate, and as the result, the reaction will include fragments that have terminated at every position where an A is required. So I guess this question is a little bit confusing in that it didn't say which ones were. The wording of this question was a little bit confusing in that this question itself had some background in that it said that there was only dideoxy ATP and not any of the other dideoxynucleotides. So that's just why it's just a random incorporation of between deoxy ATP and dideoxy ATP. So it's only focused on ATP and how, if it has an hydroxyl group or not. And in this case, there are no other dideoxy nucleotides going on. So that's how it ends up with the A at the very end, okay, of each fragment. So in a tDNA transformation of a plant with a transgene from a fungus, not found in plants, the presumptive transgenic plant does not express the expected phenotype of the transgene. How would you demonstrate that the transgene is in fact present? And how would you demonstrate that the transgene was expressed? So to do this, you would essentially isolate that DNA from the transgenic plant and probe for the presence of that transgene by southern blotting. So southern blotting is just a method that you would use to essentially see whether these transgenes are actually present, and it actually is there. Like, so depending on the size, you would see it in your image itself. In northern blotting, electrophoresis is used to resolve which biological molecules. What type of probe is used to identify the target molecules? So this is just a schematic of how northern blotting occurs. Essentially, it looks at RNA. The probe can either be DNA or RNA, but it is specifically used to look at RNA molecules, okay? So that is all I have for today. Let me know, I will have the study guide up, and let me know if you guys have any questions. Hope you guys are doing well. Until next time, see ya.